Hi everybody, this is Jason, the Science Outreach Manager and Life Science Educator here at the Children's Museum Houston. I am inside the Power Science Lab and I'm going to show you today how to make a model of DNA, and this is being brought to you by the Texas Children's Hospital. So let's give them a big round of applause for allowing me to do this easy, informative, and fun experiment. Okay, you might remember a few months back, I extracted DNA from wheat germ. And when I finally pulled all that extracted DNA out, what did it look like? It looked like a big wad of snot. You really couldn't see what DNA looked like because it's really, really small. And it wasn't until the 1950s that we actually could see DNA through powerful microscopes and we were able to know what its structure is. And its structure, when unfolded, looks a little bit like a stepladder. Now, to make this DNA model, it's really easy. Grab yourself a piece of cardstock. I chose yellow. Grab yourself some popsicle sticks or craft sticks. I chose green. Get yourself 4 different color beads and get yourself a marker. And I'll tell you how it's made. And after, I'll tell you who discovered it and why it's important. Plus, I'll show you a second way of making a DNA model that's really fun because you can twist it and stuff. So this sort of diagram DNA model right here, you can see that I have one craft stick over here, one craft stick over here. I have beads going all the way down here, and I have this dashed line right there. Let's talk about what each thing means really quickly. I'm going to set it down over here for a moment so I can sort of peek over to make sure I'm pointing at the right things. Now, if you notice, on each craft stick, I have this sort of X with dots, and then I have this sort of pentagon looking shape right over here. The backbone of DNA, which stands for deoxyribose nucleic acid, is a sugar phosphate backbone. The X right here is the phosphate, and this pentagon right here is the sugar. Now you might be thinking to yourself, is this the same sugar I eat at my table when I put it on my cereal, or if I eat a really delicious cookie? No, it's not the same sugar. Really, in chemistry, sugar is basically, in the most general sense, a crystalline structure. And when we talk about this crystalline structure as a sugar, that's why it has this very specific shape of a pentagon. Some sugars don't have this shape of a pentagon, and this is a ribose sugar. So it has 5 sides to it, okay? 1, 2, 3, 4, 5. Now, you'll notice that the phosphate right here is attached to the sugar right here, and that little attachment is called the 5 attachment, and over here it's attaching on the top at the 3. So it is interconnected at the 5 spot and the 3 spot all the way down. Now what's interesting about DNA is that it's a double helical strand when it's folded up. So this actually folds in on itself. Now you might thinking folding in means this. No, what it's actually doing is it's folding like this. Okay? It's making this double helical strand when it's folded. I'll show you that with the other model in a moment. So what that means is on this side it's going 5-3 down, but on this side it's going 5-3 up so that they can connect properly later on. Now, these beads right here, they are called base pairs, and there are 4 different types of base pairs. The orange base pair right here is adenine. The purple base pair right here is thymine. A, adenine, T, thymine, they always match up. Now, sometimes the orange will be on this side, and sometimes the orange will be on this side. And same with the purple, it'll be over here, it'll be over here. But it's always AT or TA. Now, the black one here is cytosine, and the green one here is guanine. That's C to G, and it always pairs up. So again, you can have the black on this side or the black on this side, the green on this side or the green on this side. And the way these pair up is what causes you to have your specific genetics. Now, DNA is found in every nucleus of every cell in your body, so you have a ton of it inside of you. And they are really long when you actually pull them out of the nucleus. Now remember, cells can only be seen under a microscope, and the nucleus is a small structure. It's actually one of the larger structures within the cell, but it's a smaller structure within the cell. And this has to fit inside. Guess what? You pull out one strand of DNA, it's almost 2 meters tall, which is over 6 feet tall. So it's a very long strand. So these base pairs, there's lots of coded information that gives you a specific trait. It might give you your blue eyes. It might make you left-handed. All these different things are due to DNA. Now, the way the base pairs are connected, these dashed lines, these are called hydrogen bonds. So you have a sugar phosphate backbone, you have these base pairs, 4 different types, and you have these hydrogen bonds. And that is the simple structure of DNA. DNA is one of the 4 macromolecules. It's also a polymer, which means it's a long-chain molecule with repeating subunits. Now, even though molecules are small, macromolecules are large. That's why they're called macro. That means big. Now, I said it's one of the 4. There are nucleic acids, which includes DNA and RNA. There's carbohydrates, there's lipids, which are fats, and there's proteins. When you have all 4 of those in your cells, you are conducive to be a living thing. And that is very, very important. Now, I do want to show you another way to make a DNA model, because this one right here will show you how it twists into that double helix and can fit inside your nucleus of your cells tightly packed. So these right here are little pop beads. And the pop beads are great because you have a pink and a white one, so that could be your phosphate and your sugar. The base pairs always connect to the sugar, and then you have 4 different colors here. You have red, green, yellow, and purple. Now, I haven't really decided which one is which, so the red could be adenine, and the green could be thymine, the yellow can be cytosine, and the purple can be guanine, but you can decide for yourself how you want to make it. These pop beads with the connectors here that make the hydrogen bonds can be found easily online. Now, once you've made a long strand of DNA, just take it from the top and the bottom and you can twist it. And then you can see how it makes that helical strand, and you can see how the different sugars and phosphates connect as they go, and you can just bound it up like this. And that's the tight wound that fits inside your nucleus. It's February, so it's Black History Month. And the discovery of the helical structure of DNA was aided by an African-American scientist, a geneticist named Herman Branson. In the 1940s, he was working at Caltech, which is in California, under another very famous scientist named Linus Pauling, and he helped discover the structure of protein, which helped discover the structure of DNA in the 1950s. Now, the 2 people who are credited with the discovery of DNA are Watson and Crick, but we cannot talk about the discovery of DNA without first talking about Rosalind Franklin. She did all the legwork, she used all the microscopes, and she did all the research to get to that point of discovering this double helical strand that we now call DNA. So, as you can see, it takes a lot of scientists from a lot of different backgrounds. Rosalind Franklin was English to actually discover something of this significance. Thank you for joining me and learning about how to make a DNA model. I really, really appreciate if you would hit subscribe if you're watching on YouTube. That way you can see all the videos we have to offer, our Mr. O, oh wow moments, our tater tot songs, all the other science videos we have, and our story times. Also, I would love to see what you made. So, if you're at home right now and you got some popsicle sticks and some beads and a marker and some paper, go ahead and make a DNA strand. You can make it slightly different than mine. Just make sure the sugar looks like a pentagon. Make sure you somehow signify what's the phosphate and write out which one is your base pair. So if you have a green breed for your cytosine, write that down, okay? If you do that, please share that on our social media sites. We have Facebook, we have Instagram, we have Twitter, we have it all. With that, I'm going to say goodbye for now. I hope you're having a great day, and I hope to see you soon. Thanks a lot, everybody.